So let's start very broadly with science of mental state decoding, and then we're going to talk about challenges and limitations. That's going to be a major part of the talk. And then we're going to talk very briefly about applications and ethics. So if you don't have any idea what a brain scanner is and you still want to, so that you can pick up again at the later parts. Yeah. So back in the early 2000s was a time where people kind of began to get interested in more sophisticated forms of data analysis of fMRI data. And people previously had been doing pretty, I'd say, rudimentary analyses where you apply some statistical model to each voxel, one voxel at a time. So if you do that, you get some interesting information about the relationships between single voxels and, say, cognitive variables and treatments, etc. But it kind of doesn't give you the picture of how information is coded in ensembles of activity measures in brain areas. So multivariate approaches, decoding, regression approaches came up in the kind of early 2000s, and I was at the time in London, and we also started doing that kind of work. And so it was quite a different approach, because now you're asking not, can I prove that a certain part of the brain, a voxel, is kind of driven by some task variable, but you're asking the inverse question: what can I infer about a task variable from all sets of brain activity measures? And so this is something that we then applied to fMRI signals, as did other groups, and it's kind of gone quite a few different ways, with representational similarity analysis playing a big role now. People using more sophisticated classification approaches and asking whether the kind of like properties of deep neural networks can be mapped onto brain signals, etc. So this kind of was kind of like petering along, and there were kind of quite some interesting developments. But then back in 17, 18, there were these big announcements from high-tech companies claiming that now they're going to invest heavily in brain scanning technology and in kind of brain reading, the science of decoding mental states from brain activity. Most notably, Elon Musk was very invested in that with his Neuralink, and Facebook was very invested in that. And there were all these claims that were being made, like, we're going to build a system that is non-invasive, and you can use it to type a letter, and it's going to be faster than if you're an experienced typewriter, and things like that. And if you were familiar with the science behind this, it was quite clear that a lot of these claims were obviously overblown, and it wasn't really clear how you would end up being able to get that kind of a signal out of the brain. I think the biggest challenge we have in mental state decoding is just getting decent signals from healthy humans, obviously. And there's no kind of magic bullet that we've found so far. But there have been some interesting developments. For example, about a year ago, Elon Musk, with a lot of kind of public attention for implanting his Neuralink recording devices into pigs. And so I'd say, I wouldn't say that whatever they found that was revolutionary, but the technique is at least kind of interesting when you see what can kind of happen in a field if you get people with very different expertise to come together, like materials science and recording and data analysis and cognitive science and so on. So there's a lot of interesting kind of movement there. But I still think that we need to kind of think about what are the big challenges in this field? What makes it really difficult to decode mental states? And the reason I like to think about challenges is because they should define what you're going to be working on next. I mean, I would say don't work on something that you know how to solve, but work on something that is a big challenge and is kind of really difficult, where you have to kind of spend a lot of time thinking about it. And of course, in the wake of this, there were also some interesting exuberances. For example, this is, I think, from The Guardian, and it says a mind-reading device can answer questions in your head by picking up signals from your brain when you think about saying something. This is the alter ego device, and yeah, fair enough. I mean, but it's definitely not picking up signals from your brain. First of all, look where it's placed. You hang it over your ear, and what it's actually doing is recording EMG from your jaw muscles. So what you have to do to operate the system is not generate something in your brain with a brain-computer interface. But what you're actually doing is you're kind of subliminally speaking, activating your jaw muscles, and that's the way this is read out. And I'd say fair enough. I mean, it's an interesting system because typically you might just not want to speak in an application situation, but it's much easier to get these kind of signals if you have an effector that is still active. Yeah, so you might not want to have to record from inside the brain if you can record the effects of something inside the brain on peripheral muscles. So it might go just as far. No, there seems to be a lot of confusion about what can be done. And there also seems to be a lot of confusion about what actually a technique is that really relies on brain signals. And in the brain-computer interface community, that's exactly quite the same, because often what these brain-computer interfaces are recording is where you're looking. So BCI signals often allow you to gaze freely, and they have whatever, some frequency markers or something like that. And what you're recording then is ultimately just where you're looking. And they're much easier to devices to record where you're looking than just putting on a brain cap, so an EEG cap or something. So I'd say, yeah, I mean, there's a lot of interest, but it's also, some of it is kind of a little bit kind of meager in its kind of scientific content. I don't mean this here. I think this is an interesting system. It's just not about the brain. So really, in a nutshell, what are we looking for? So this is a slide to infuriate philosophers, particularly dualistic philosophers. They always get very angry when I simplify this to this degree, but I think the way we should be thinking about the brain is similar to a CD. And I don't mean in terms of its dynamics. I mean, the brain is an active dynamic system, obviously. But if we want to understand how our thoughts are encoded in our brain, it's similar to the way a piece of music is encoded on the surface of a CD. Yeah, so it's the pattern of the pits that are on the surface of the CD that code the information about the music. And if you have one piece of music, you have one pattern. If you have a different piece of music, you have a different pattern. So there's a relationship between the code and what it's representing. If you break the CD, you lose the music. It's the same with the brain. If you break your brain, you die, for example, then you don't have your experiences anymore. So I think this is a very crude analogy just to bring home the idea that the brain, the way I think about it, is like a carrier of contents. And I don't want to get into kind of detailed philosophical arguments here. No, I don't think the brain is a CD. But I think it operates, the way information is stored can be understood in very similar terms. Big problem is obviously that you can look in the some IEEE journal to find out the way the CD player works, but there's no IEEE journal paper that explains to you how to infer the brain signals, how to analyze the brain signals in order to infer what a person is currently experiencing. So I'm just going to spend a very brief amount of time on that, saying that I don't really think that we understand what lay people refer to as a language of the brain. And this whole field is called brain reading or mind reading, and it's got nothing to do with reading at all, because what ultimately people are doing here is using brute force decoding techniques to infer something about a person's mental states. This got nothing to do with understanding the syntax and semantics of kind of some kind of language in the brain. So I think brain reading is a really bad term because of the reading part of it. We're not really reading in any sense. It's not that systematic as reading would be. So let's look at the simple problem. So let's just, this here on the left is a picture, a sketch by Ernst Mach von Analyse der Gesichtsempfindung. And it's his attempt to capture what our experiences are while he is looking out of his left eye and on some afternoon sometime in the late 19th century. And let's just focus on one aspect here. For example, the brightness experiences he has here in this region of the visual field. Now, the brightness sensations there can vary. So they vary along the dimension of intensity. So you have different conscious sensations. And if you want to explain what a person is experiencing by these brain signals, you want to have one state of the neural carrier that uniquely encodes what the person is experiencing. So this is just kind of like the neural carrier, some property, for example, signal intensity in V1, or spiking activity of V1 cells, luxotonic V1 cells. And they might, for example, when you see something that's bright, they have a high spiking rate, and when they kind of see something dark, they have a low spike rate. This simplification, that's not quite the way it works. It's a kind of like bipolar system, the way light intensity is coded. But that's the kind of thing you're looking for. You want to have a situation where for every experience you have a specific brain signature. And There are intensity codes in the brain. For example, for brightness, it seems that brightness is encoded with intensity signals. But there are two main kinds of signals, I would say. One is you could decode the sensations also in a labeled line code, or what I call a sparse multivariate code, where you have one unit representing every experience. So let's just say you've got 6 different brightness experiences. You'd have 6 different neurons that are encoding that specific brightness experience. Now, the labeled line code is very inefficient when it comes to intensities, but it has one big advantage, which is that you can read out every individual line has its own meaning. That's a very nice thing about the labeled line code. So if this, for example, would be different objects that a person is experiencing here on the left here, then you could code every object with a labeled line dedicated to seeing whatever, a face, a house, a mouse, a cat, a dog, or whatever. And there are situations in the brain where information is coded roughly in that way. For example, the somatotopic maps and somatosensory and motor cortex, these homunculi maps, they're basically labeled lines. Yeah, basically every neuron is dedicated to one place on the surface of your skin or one specific effector. That's basically a labeled line code. And ultimately, the analogy is that you can kind of think about this like one-to-one mapping. Now, there's another kind of code, which is a distributed multivariate code. And the distributed multivariate code is now encoding every one of these states you want to encode in a population signal. So the difference here is that you can't interpret a single line anymore. You can only interpret the set of activity values. And now, in order to understand what a person is experiencing, it's not enough to just look at one single unit, but you have to look at an ensemble of units in the neural carrier. And we have examples of that as well. For example, population tuning of orientation signals follows that principle. It's not just, people always think of these tuning functions as representing one specific orientation. The cell is tuned to one orientation. Well, if you look at a Fisher information analysis, we know that the cell, for example, signals most information about the sidebands of its tuning function, and not so much on these flatter parts of the tuning function. And if you look at object encoding in the brain, this is now kind of a snapshot of larger region of visual cortex. That seems to follow this principle as well. So we have these distributed multivariate codes as well. Now, the next question you can ask is, well, what is the property that these channels have? And I'm being very generic here, because you could say, well, it could be spike rates in multiple cells, but it could also be local field potentials recorded at a local area. When you have quite some flexibility how you define the channels here, but the basic principle is very similar. And then what you do, let's just say you have a person looking at the Brandenburg gate or a bouquet of roses here in the top right. And I'm not going to spend much time on this slide. If you want me to, I can explain it in detail, but I'm assuming most of you have a basic understanding of classification techniques. So the idea is that every, for example, object that you want to decode has some multi-voxel activation vector. And if you just, for example, look at the first 2 dimensions, X and Y, you can think of them points in a 2-dimensional space. And then the multiple recordings, say, for example, of this experience and of that experience, just give you a cloud of measurement points. And you can dissect them in various ways using different classification approaches. For example, linear, nonlinear classification, et cetera, et cetera. So, let me know if you want me to spend time in explaining this slide in more detail, but I'm assuming that most of you know this stuff already. And then the question is, where does the patterning of the brain signal come from? And there are different ideas. So one idea is that if you think about orientation columns in early visual cortex, that they are kind of like their spatial scale is smaller than the size of a typical voxel, but due to the fluctuations in the density of different cell types with different tuning profiles, you get a subtle patterning of these fMRI voxel responses, and that's what you can use for the classification also of kind of contents that are coded at a scale that is smaller than the typical resolution of your voxel. And there's an interesting kind of pointer that you can set here to ultra-high field scanning. Say, for example, if you reduce the voxel size to 1 millimeter or even sub-millimeter, you're ultimately still doing something quite similar because you're never measuring a single neuron. So the smaller you go with your voxels, it's never the case that you really record only a really pure signal from only one orientation column or something like that. In fact, even if you do voltage sensitive dye imaging, you don't get that. You don't get these pure, pristine signals that reflect only operation of one specific cell type. So even if you kind of work to go invasive and you were to look at these signals, they're actually much more complex than you might think at first sight. So there's another idea that some of the patterning that we see has to do with macroscopic biases. This is the work from Freeman and colleagues from New York. And I guess at the end of the day, what we know is that ultimately the voxel is sampling neurons, and the neurons are distributed kind of like a collection of different properties within a voxel. You can't resolve the kind of tuning functions of individual neurons this way. So you're going to catch in your kind of voxel, you're just going to catch a large number of different tuning profiles. And the best you can do is, as you say, you have some kind of information. But if you want to then say what specifically it is at the neuro single cell level, I think that, I mean, it's quite tricky to tease out that kind of information using fMRI, unless you do some modeling. We've done that in the past, but even then you make all sorts of assumptions, and the question is whether kind of this is valid or not. So we've used this in the past, so we don't just work on vision. We also work on executive functions, and a study that kind of some of you might have already heard about. And I'm just showing this to those who kind of might not have a background in these details of kind of classification analysis. They got people to make spontaneous decisions between left and right button presses, and to remember a letter was on the screen when they made up their mind. And then we used a classification approach, scanning throughout the brain for information about the upcoming choice. So this is basically where you can decode the choice even before a person believes to have made up their mind. And what you can see is that the voxel pattern is different depending on whether a person is going to choose to move their left or right, even before they themselves believe they've made up their mind which hand they're going to move. Another thing you can see is the accuracies are very low. We'll come back to that again, because while they're significantly above chance, the question is, well, what does that really mean that you get these kind of low classification accuracies? And the field of fMRI classification, you often live with these kind of rather meager classification accuracies. And one of the questions, of course, what does that mean? And also, what does it mean if it's not 60, but it's 40%, where 50% is chance accuracy? Yeah, so what if you systematically get below chance accuracies? I mean, this is something that people don't normally find, because the way they do the analysis of their classification maps is that they look for regions where you have above chance information. You should always do the sanity check and look inverse. Where do you have below chance information about your classification? Because that presumably says that you're doing something wrong, or it tells you something interesting about the conditions that you have. For example, the way information is coded in the brain. Okay, I'm going to skip over this. So let me just come to the second part, to the challenges and limitations. This is what I want to spend most of my time on here. And so I mentioned this already, that we're sampling the brain with voxels, and they're kind of capturing only kind of a very macroscopic picture of brain activity. I like the term brain activity, actually, because it's so vague. You don't have to say inhibitory, excitatory, spiking, dendritic, whatever. I mean, you just say activity. And I think of this as something a little bit like a seismograph, where you're just picking up something is going on here, and then you try and interpret that. So if you look at an fMRI voxel, it can have up to 1 million neurons. So that's quite a lot of neurons. And most of you know this. So the question is, well, what about ultra high field MRI? Can we go finer? Can we, for example, do columns like orientation columns or eye dominance columns, or can we even look at layers? And there's a lot of interest in this at the moment, obviously, and there's some, like Huber, for example, doing really great work, or Niklas Weisskopf in Leipzig. So there are a number of groups doing great work on specifically layer-specific imaging, but it's not that easy, because ultimately you have a common vasculature. If you look at the different kind of layers here, you have a common vasculature. So basically, if the vascular penetrates from the surface to the depth and drains to the surface, you can have it like a common hemodynamic component that's kind of like going to lead to artifactual correlations between these different layers. And you then have to do modeling assumptions in order to kind of interpret the findings. So it's not that easy. Even if you get the resolution that in theory your voxel is small enough to pick up just one of these layers, it's quite tricky. And in fact, it's quite the same with the, say, orientation columns in V1. And it's been really tricky. I mean, some groups have been managed to do this quite well, but we've been working on this now for 20 years. I mean, since I started with fMRI, people have been talking about this, but it's not like something that you could routinely use and everybody now is studying cortical columns. So this is something that is kind of, I'm really enthusiastic about it. And I love trying, and I'd love to continue trying, but we should also face the fact that possibly where fMRI is not going to be the technique where we're going to be able to record pristine separated signals. So, because the problem is, you can get these maps, for example, these orientation maps here, but they're obtained by differential imaging. So what you do is you compare what is, for example, the most strongly activating orientation in this map, but you don't get a region that responds exclusively to one orientation or another orientation. So you have these kind of dependencies. And in fact, if you do optical imaging with voltage-sensitive dyes, it's very similar. You don't get these pristine signals. But if you want to, for example, do a perception study where you say, well, I'm going to look at what simultaneous brightness contrast, where I have a gray surface and it's either on a white or on a black background, then, well, you're not going to be able to very easily study the center independently of studying the kind of direct effects of the background. So it's kind of quite tricky because you can't measure really selective signals. You always get a signal that's contaminated by things. So that's kind of one of the challenges that I would see. And so one of the things that might be possible here is to actually look at voxel statistics. So we might want to move away from maps and layers, and certainly when we go to finer resolution, I might just kind of go back to the idea of looking at the ensemble statistics of voxels. For example, this was first study we published on multivariate classification back in 2005. And this was a study where we just presented two orientations and measured, and every voxel measured an orientation tuning profile. And then we just compared this to chance. But you can look at the entire population statistics. So possibly a way to think about this, which is also a problem in electrophysiology if you do multi-unit recording, is you're just drowning in all the possibilities, the different tunings that these kind of units have. And possibly population statistics, population properties are a better way forward than to just look at, say, for example, single voxels or just the classifier output. I think there's a lot of information buried that we don't normally look at when we look at the kind of populations of voxels, not a classifier output, but looking at more in detail at the kind of the properties of kind of like the ensemble of single voxels. Now, another challenge is the individuality of the kind of way information is coded in individual brains. So, for example, what you can see in the top here is the famous fusiform face area by Nancy Canvisher. Yeah, that's roughly in the same place across different brains, not completely. So there's no perfect overlap. So even there, it's quite difficult. You train a classifier on one person, it's not going to that easily generalize to another person. It does to some degree. And if you look at these orientation maps, it gets even worse. So this is Kavitani and Tong from 2005. You can see the different colors correspond to different orientations, the kind of preferred tuning of individual voxels. Every one of these little segments here is a voxel. And you can see that there's not that much similarity from person to person. And as a rule of thumb, I mean, people have asked me this many times, and my kind of rule of thumb is that processes should be more similar, contents should be more different. So basically, if you want to know what specific individual it is, or what picture exactly you're looking at here, then the patterns might be quite different from person to person, just due to the way in which every individual brain follows its own developmental trajectory. And part of this, as we know from the work, among others, from Wolf Singer, is self-organizational, the way the visual system calibrates itself. So we expect there to be some differences between people in the way exactly every unit codes for specific properties. So that should be quite individual, but processes, for example, whatever, top-down attention, you have FEF modulating, say, V2 or something, that's something where you might see more similarity across people. So if we go down to contents, I think you need to go to individual people if you want to kind of get a decent accuracy out of it. But individuality comes even more extreme. The reason I'm mentioning this is because we're also involved in using classification and machine learning approaches to data from diseased brains. So, for example, we looked at multiple sclerosis and Alzheimer's, and as everyone has, and looking for kind of techniques in which you can do good classification or kind of progression of the disease, like when are you likely to convert from minimal cognitive impairment to full-blown Alzheimer's diagnosis, these kind of things. So the thing is that the brain is so tremendously plastic. So this is a famous example from 1980. And there are other examples that are similar, not quite so spectacular. So this is on the left is a CT from a healthy brain, and on the right from a hydrocephalic brain. And this was discovered by chance. And this guy here, whose brain is depicted on the right, was studying math. So he was a student at university studying math. So, and if you look at the kind of fluid-filled cavities here, the ventricles, they're massive. So I think it's going to be very difficult to generalize from the left to the right. So if we go from healthy humans, the kind of brains are reasonably similar, reasonably, not that similar, but reasonably similar. But the disease can take so many different forms that, I mean, every one of these somewhat has a bit of a unicorn property. So the kind of every individual brain lesion and brain damage is so different that it might be quite difficult to study the information encoding properties on the left brain and transfer it to a single brain of which there's only one instance. So training is already quite difficult if you have a sample of one brain and transfer it to the right. But I mean, obviously, this is something where I think what's happening right now is people are realizing the necessity of kind of big databases, and the successful kind of, I think, next generation disease classification applications are not going to be, hey, I've got my handcrafted, hand-selected group of 30 patients and 30 controls, but it's more like 1,000 plus, and just skimming off data from kind of some startups skimming up data from kind of like many hospitals, data acquired under many different conditions, and combining it with deep learning approaches to kind of learn the basis functions with which you can encode the kind of the brain structure. So I think this is good, but this means also if you're a kind of small unit at a university hospital and you want to say, well, I get my 20 patients and my 20 controls, then machine learning, I think, is going to be a bit difficult for you in the future, because I think the future is to really collect lots and lots and lots of data. So another question is, and this is now the kind of simple explanatory slide, is like, well, do I have to learn every individual thought that I want to decode? And this is from a kind of Scientific American paper I did over 10 years ago, but it illustrates the point. So let's just say you have different thoughts, and every one of them you've measured the pattern of brain activity. Okay, there's a 30-second break. My son is just coming back home. Something. Okay, sorry about that. Sorry about that. So it's childcare. So the idea is that you have these different thoughts, and they're associated with unique patterns of brain activity. And let's just say you've measured this because you had the person in the scanner thinking one thing or another, for example, thinking about a car or thinking about a bicycle, and you have the pattern of brain activity associated with it. But now you've observed something you haven't observed before. The question is, what could this be? And the example is chosen in a way that what this is a mixture between the pattern of brain activity for a car or pattern of brain activity for a bike. And so you might as well guess that this is actually a motorbike, or a bicycle on top of a car, or a bicycle accident with a car, or something like that. Something to do with bikes and cars. And so this principle of kind of mixing or interpolation works to some degree. And it doesn't work very well, but it works to some degree. It's not that it doesn't work. Like if you're, for example, into semantics, there is this issue of compositionality, and there is some compositionality in here, but it's not really that perfect. Yeah. So, and that's why that's a basic assumption that you have for lots of modeling approaches. So if you don't want to measure every individual mental state and the corresponding brain activity patterns, you can use models to simplify this. And for example, this has been done for semantics by Mitchell, 2008, and for kind of YouTube video sequences with dynamic stimuli by Nishimoto and Gallant, and this famous YouTube sequences here. And so every one of these needs a mathematical model to cope with the huge complexity, or kind of not complexity, but the kind of dimensionality of the space in which you want to do your decoding. And let me just kind of give you, sorry, this is where I want to go. So, and I just want to provide an intuition for people who might be here who are not that clear about this problem of dimensionality. So this even comes about when you're looking at receptive fields. So you might have heard in the cognitive neuroscience basic course on vision that Hubel and Wiesel, they were kind of like looking for the tuning properties of cells in V1, and they couldn't really find anything. And then one day they put in a slide that had a crack in it, and they found that the cell responded to this slide with the crack. And then they thought, well, it might not be point light stimuli we should be looking for, but these kind of elongated line elements. Well, yes, but what if they'd done something else? So there are so many different stimuli that you could have used. Perhaps the whole story of V1, I don't think that's true, but you could be kind of led devil's advocate. Well, what if just the proper stimulus hasn't been found yet? And this fundamental skepticism has kind of plagued people in vision science for a while, but I just want to provide you with an intuition of this. So let's just say we simplify the problem of finding out what a voxel is responding to a problem of 10 by 10 grid of black and white elements. So now we're really reducing this. Let's just say you had eyes that consist of 10 by 10 receptors that either say black or white. And let's just say that this is our vision science little micro world. And you could, most of you would be able to say, well, how many different images there are, 2 to the power 100. And it doesn't sound very much, 2 to the power 100. But actually, if you bring it in perspective, so that's roughly 10 to the power 30. So if you could start at the beginning of the universe and measure one brain response to one image every second, you would still have to do this 10 to the power 13 times longer than we've already done it since the beginning of the universe. I just want to be clear here that even the 10 by 10, so 100 cells, black and white, the dimensionality is just unbelievable. And I think this is you can't just think that you're going to be able to stumble through this space and stumble on the right stimulus. And in fact, you have a very similar problem when you're trying to identify mental states, because what you want to do is you want to find for each mental state, you want to find the corresponding brain signature. And even for pictures, the dimensionality is just so absurdly huge that you can never even dream of searching exhaustively the whole space. It's completely impossible. You just have to realize that we're never going to be able to do that. Humans are never going to be able to do that, search this whole space. And we wouldn't even have enough humans available as participants to search this space, I guess, because, within a lifespan, you wouldn't even be able to do this of a normal human. So we're going to have to live with simplifications, and the mathematical models obviously give you simplifications. And another place where you can get simplifications from is simply priors. This is now from the work, a nice paper by Schoonmakers and Van Gerven from 2013. And what you can see here is they were decoding handwritten digits from signals in the human brain. And you can see in the top row is the original digits. And then you can see the reconstruction, the maximum posterior estimate in the bottom row. And it looks good. Yeah, it looks good. In the second row, what you have there is the maximum likelihood estimate. And the maximum likelihood estimate is really what the brain gives you. Yeah? Not the prior, it's what the brain gives you. And it looks good in the bottom row, but the way it looks good is because you have to combine the maximum likelihood estimate, which is what the brain gives you, with the prior, which is basically something like the average across all these different digits. And what you can see here is that if you just go by the brain signals, there's not that much in there. And similarly, if you go to a couple of papers, for example, from Jack Gallen's group, that do Bayesian analysis, Bayesian reconstruction, if you look at the way the Bayesianism is implemented there, is it's stick functions where the set of images that are the ones it can be are basically there in the set of possible stimuli, and the others are not. So that's why you get these nice reconstructions often. But if you look carefully, if you look at the maximum likelihood estimates, they often don't look that fantastic as these nice videos and pictures look. So again, I think we have to be careful that we don't overestimate, even for visual stimuli, where we've got such a large part of the human brain dedicated to analyzing visual stimuli. Even there, we've got so much real estate in the brain that we could sample. Even there, our ability to reconstruct images is not really that fantastic. Yeah. And we've tried maximum likelihood estimates on various naturalistic stimuli and things like that, and you don't get very much. And if you look on these papers, the kind of maximum likelihood estimates, they really don't look very good. I mean, you can't tell anything at all. It's just like salt and pepper.
One question about this.
Go ahead. Yeah, please. I said interrupt me if you want.
Yeah. For maximum likelihood estimates, you usually assume some density of your brain data, meaning that you already make an assumption about the distribution.
Yeah.
So assuming that you find the, let's call it correct, or a very suitable distribution, let's say one with a very small Kullback-Leibler divergence, could you then increase the signal for a readout?
So we've spent a lot of time with this recently, and that's a couple of slides that I skipped over because we've been looking in Gaussian process regression and combining maximum likelihood estimates then for voxels. And then the question is basically, how do you estimate the density function? I mean, I can go back to that. I skipped over it. So these were stimuli where we had these coherent motion stimuli on for 2 seconds, and then people had to make a judgment which direction they were drifting in. And the idea of this study was that most of kind of perceptual decision making is discrete. So let's say left versus right. And what you often have there is a confound between your stimulus and your decision and your motor output. So they're kind of like, they're very hard to disentangle unless it's 0% coherency. So what we'll be doing here is Gaussian process regression. And what you do there is basically you have different trials with different directions. You have an ensemble of voxels, but if you just look at one single voxel, you're estimating these tuning functions. And they're basically, all you do is they're basically, you make a assumption on the smoothness with which the tuning functions change. So every one of these is a kind of likelihood function here. Every vertical in this is a likelihood function, and it's a likelihood function as a function of the direction of motion of the stimulus. And then you basically, Gaussian process regression estimates this. And this actually allows you to do a little bit better, but it's not that you suddenly do spectacularly better using this kind of approach, yeah? And you estimate the smoothness from the data and things like that, but I wasn't sure if kind of wanted to go into that technical level of detail. Okay, so let's just go back here. Now, another challenge is what I would like to call label confusion. And this comes out in an area we're working in my research group on visual working memory with my postdoc, who is now actually has his own Eminota group, Thomas Christoffel. We're working on encoding of visual working memory contents for a while now. And so a typical scenario is, let's just say you're kind of memorizing, say, for example, a lion versus an elephant, and you find information about the lion versus the elephant with a decoding approach in the sensory cortex, but also in lateral prefrontal cortex. This is counterfactual because you don't really find it in lateral prefrontal cortex. It should be there, but you don't find it. We tried, I mean, it's very hard to find sensory encoding in lateral prefrontal cortex in human fMRI signals. Now, you could say, well, the information is in sensory cortex, and there's a second representation of the elephant versus lion or kind of animals or something like that in lateral prefrontal cortex. But what you often don't talk about, and this is actually a big problem, is that they could be doing very different things. For example, sensory cortex might be encoding the difference between lions and elephants, whereas lateral prefrontal cortex is just setting you up for some kind of discrimination you're going to have to make, because if you are expected to memorize an elephant or a lion across a delay period, you're not just retrospectively really coding the information that you previously needed looking back, but you're also preparing yourself for the task that you have to do, which is select the stimulus that it was among, say, 2 or 4 alternative stimuli that get presented later. So it's not just retrospective information, it could be prospective information. It's guiding your attention in order to solve the upcoming task. And we know that there are feature-based attention signals there. Also, spatial attention. It's quite well known that every image comes with a different distribution of spatial attention. So when you're looking at a bicycle or a car or a lion or an elephant, every time your spatial attention distribution is different. And we know that there are spatial attention signals in these areas. So possibly what you're encoding there or decoding there, the information, has got nothing to do with the semantic properties, but it might have to do with the color of the feature-based attention signature that you're looking for, or spatial attention, abstract categories, you name it. So I'm just mentioning this here because when we do classification, we're always in this forward-thinking mode. We say, well, these are my experimental conditions, and I see that I can reconstruct these experimental conditions or decode them from signals in the brain in that area. So that area must be encoding this. But if you don't just have to look at the brain signals, you have to also think about what are the other cognitive variables that might be driving this instead, this effect. Yeah. And if you start looking, you will find, and there are many. So the tricky part is really kind of coming up with these kind of alternate explanations and doing experiments to rule them out. So you can see this is kind of like a long list that I'm going through here, but I thought I just kind of use the opportunity to give you a slightly different story today. Another question is like, what does classification accuracy mean? If you get an accuracy out of a classifier, say, again, this first study we ever did, just with distinguishing between different orientation stimuli, and you might get an accuracy of 80%. What does that mean? Well, there are many problems here. You might be underestimating information. And the reason you might be underestimating information is because your voxel just bundles together so many different cells. You might be overestimating information. How could that be? Well, because the voxel integrates across a period of time, and the integration time period of the voxel might be much longer than the integration time period at the neural level. Yeah, say up to a second for these coherent motion stimuli. And so you might also be overestimating the information, because ultimately the bold signal is not what's processing the information, but the neurons are processing the information. And you can't that easily compare information across areas either, because you have different number of voxels, different distribution of cells, different sampling patterns, different signal to noise ratios, etc., etc., etc. Different hemodynamic efficiencies, to use that label. And also it depends on the way you partition data into training and test and things like that. So there's so many, so you often see people saying, well, 55% is not a good accuracy. Why don't you get 75%? Or 52%. Well, I think that's not so good comment, because if in your voxel you have only 5% of the cells participating in a certain operation, then you can't really have that easy effect size assumptions, what you're looking for in these voxels. I think that's actually a little bit tricky. So how are we for time? We got like another 5 minutes or something?
Yeah, 5 minutes should be well.
Okay,
just feasible.
Make a jump here. We'll come to 2 more points, and that is negative or below chance classification accuracies. So this is something that my postdoc now, Kai Gergen, spent a lot of time staring this problem down, and that is interactions between, for example, the way in which you classically control for an experiment using counterbalancing, and the way you do data analysis with cross-validation. The problem is basically, let's just assume that you counterbalance your data. So you're running an experiment where you're first showing whatever, a face, and second you're showing a house for 1 minute face, 1 minute house. And you're counterbalancing it where you're first showing the house and then the face. And you do this a couple of times. Now, when you counterbalance, that is the reason you counterbalance. The stimulus order shouldn't play a role. But when you then do this in combination with cross-validation, for example, then you say have 4 runs, and your 4th one is your validation data set, and the other 3 are training data sets. This classifier can pick up order effects because you're removing, by using the cross-validation, you're breaking up the counterbalancing again. And that way you can actually get negative classification or below chance classification accuracies. And if you start looking for them, you see them all over the place. So what Kai has spent a lot of time with, and he actually published a toolbox called the Same Analysis Approach Toolbox, is to test your design variables, for example, what conditions you have, a priori before you do the experiment, to see whether they would suffer from this problem when you then later on do the scanning and do the data analysis. So basically looking for the artifacts contained in your design. And so it's like a vetting process of experimental designs that you can do in this way. So again, I think, if I go back to when I started using machine learning techniques in fMRI, and what happened was I would then travel around and visit some labs, and people would say, oh yeah, you got 80% classification accuracy, but we get 99%. And you go and talk about to them and you realize that there's some circularity in the kind of way they divide up training and test data set. So people are still doing these mistakes today, and I detect them regularly, but these kind of mistakes here are virtually unknown. People don't even think about this. Yeah. So I don't mean to kind of be so gloomy here, but I mean, use every one of these as a pointer just for something to kind of be careful of when you're using these kind of approaches. And finally, I'd just like to point out that when you're doing brain reading or mental state decoding, you are ultimately, you need a training signal, and typically from the participant. And so if you want to give a label like, do you like this or not, some kind of very subjective variable, you're going to need phenomenology by the participants. They get, or pain ratings, for example, is another example. So like a really clear example. So it's like, it's only going to be as good as your phenomenology. And my prediction is that if we look in the future of 50 or 100 years, we might get to the point where the brain recordings are not the limiting factor, but the phenomenology that people tell us what they really experience in a certain situation. It might actually turn out that the psychological challenge is much larger than the brain challenge. Just looking forward. Okay, I think in the interest of time, I'm just going to stop here and going to skip the issue of the applications. But or do you want me to just go on for 2 or 3 minutes, just talk about the applications very briefly? It's up to you.
I think 2 to 3 minutes is fine.
Okay, so just briefly talking about applications, going back to the beginning. So what I tried to present you with are a number of challenges we have in our research field. And they're challenges with respect to how well we can identify mental states from brain signals, but also how, as a field, we do the research and what kind of blind spots we have in this. Now, there are a number of applications that people are really interested in today, brain-to-computer interfaces or detecting consciousness and vegetative state, but also all this neuromarketing and forensics that are kind of like very en vogue. And I'm actually quite outspoken that I think that these techniques don't really do what they claim to do. I don't think that lie detection or neuromarketing is in a stage at the moment where you can claim that these techniques really do what they're claiming to do. And we could spend a whole talk just going over that. I think ultimately what we need is something like this NASA systematic that was also adopted by the European Space Agency later. It's like a technology readiness level. So possibly whenever people talk to the media about their research, we want to communicate a technology readiness level. So the very basic down technology readiness level 1 is someone claims that someone found something in their lab, but they're not quite sure. And the top would be, you have an operative system like a cell phone that works, and that will kind of be technology readiness level 9. And the reason the NASA introduced this was because they got up to, say, technology level 5 or 4 or 5, and they discovered that it's just such a long haul to go from there to something that really works. So even if you've got your demonstrators and so on, the most money gets invested in the upper stages. So I think we have to bear that in mind whenever we look into this field and we look at, for example, high-tech companies claiming that in a few years we're going to be able to have these turnkey systems that can solve all these everyday problems with brain reading. I think we need to communicate this very clearly. And I think in terms of neuromarketing and lie detection, for example, this has been going around for over 10 years. And still, I wonder if there is any basic content in there that is worth using. These systems are not really very well validated. So I'll stop there with this critical word. And sorry for this gloomy talk. But I hope you kind of were able to pick up some bits and pieces that you found interesting. Thank you.
Great. Thank you very much for this interesting talk. We have several questions in the chat already with the persons who asked.
I stop the presentation so we can see each other, right? Better.
Yeah, I think that's conducive. Okay, so Ritu, would you like to ask your question personally, or should I read it out?
I can go ahead if you can hear me. Perfect. So thank you for the very interesting talk. I had a question relating to the prediction of the choice that the person would make was left versus right. So which brain regions showed the predictive choice signal? And on a related note, I was just curious to know how can you image the cerebellum as well as the other brain regions, because the cerebellum is quite folded. So yeah, these two questions.
Yeah, I mean, you can image the cerebellum, but for some reason, it's very rare. I mean, we don't mask it out. It's just we don't really see very much there. That might have to do with voxel size. Or with the specificalities of perfusion and so on. So we don't really see the cerebellum very much. And there are cognitive deficits if you lose your cerebellum, but it's not like you can't think or can't make any decision anymore. So, I mean, so that's one part. And what was the other one? Oh, yeah, the regions, posterior cingulate, precuneus, and frontopolar cortex. And if you can say something like that, it does make sense, because frontopolar cortex has been repeatedly shown to be involved in what's called prospective memory. So basically where you store plans for things you want to do in the future. So it does make sense, but the detailed, I mean, we spend a lot of time looking at task encoding. So these are endogenous tasks. The reason we started with these endogenous tasks was mainly because we didn't want there to be an artifact of the queue. That's the reason we started with these self-chosen tasks, but then they turned out to be an interesting question in itself. But there's like a whole network of regions if you're not just interested in this kind of long-term perspective information, but kind of like the kind of more immediate encoding of task sets is along the medial wall and the lateral prefrontal cortex. And the way kind of the architecture is not that well understood, I'd say, today. Yeah.
And how do you separate left arm movement versus right arm movement from left choice and right choice?
Oh, the motor cortex comes in much later. I just basically used that. Sorry, I skipped over that because I wanted to talk about this gloomy stuff.
Okay.
So I skipped over the details of these experiments because, like, if somebody talks about these experiments, I feel like feels like there's no single molecule in the world that hasn't been confronted with me talking about these experiments so far. So I apologize for not going into the full details. I felt like I don't want to keep repeating myself every time I talk to people in Tübingen. So the answer is that the motor cortex kicks in much later. Yeah.
Okay.
So you could decode from motor cortex, but much later. Yeah.
Okay. Thank you.
Okay. Next quick question from the chat. Do you think that all mental states such as pain, joy, etc., can be reduced to electrical signal of neurons or molecules like neurotransmitters?
There's a lot of complexity in that question and hidden depth. So it's very easy to answer it just like yes or no. So I think that we haven't been able to study the brain insufficient detail so far in a living human in order to say what, apart from fMRI voxels, EEG channels, some invasive recordings of population signals, for example, or a few lucky spikes we got in medial temporal cortex, where we've only been able to record single cells in humans so far, mostly, 99%, that we can't really say what the exact, I'd say, biophysical aspect is that is kind of most closely related to the encoding of the content. So that's one way of looking at this question, is to say, well, there are many possibilities of what it might be, and we are only looking at those that we can pick up with our imaging technology. The second way to look at this question is whether, because you said reduced to, I think that's, I mean, I don't mean to be critical here, but I think about the problem in a different way. So there's all this debate about consciousness and whether it's caused by the brain or it's a different property of brain signals or whatever. And you can talk about that until the cows come home. There's no point in making these discussions. What we can do as empirical scientists is just observe the common occurrence of things. And what we can observe is every time the person has this experience, I observe these things in their brain. If that means the brain has caused the experience, or the experience is a property of the brain, or an emergent property, or well, you can study that, but like a epiphenomenal property, often if you go into the details and you really spell it out, you can't really answer these kind of questions with data. Often these are metaphysical questions. And so I just think all we have to do is just establish the mapping between the brain states and our experiences, and that's all we can do. And I think that's already a good thing. If we can achieve that, then we've already gotten quite far. And then we can leave all the speculation to people who like to go beyond the data and go to metaphysics. I'm happy to for that to be done by people with a different profession.
So I think we can squeeze in one very quick question. Sophia, do you have one?
I think Shervin was first.
Okay, then Shervin, maybe.
Thanks for the great presentation. I wanted to ask you, I mean, given that I think neither with the fMRI we can go to the individual units, and with invasive recordings we cannot capture the whole brain. Do you think fusing signals is a promising avenue? And if you think yes, what is the first step you think should be done?
Sorry, the sound quality on my side was very poor. Could you just repeat that? Sorry, I just acoustically couldn't understand it.
Can you hear me now? Better?
Yeah, much better. Yeah.
Okay. I was asking, I mean, given that with the fMRI, we cannot go down to the individual units, and with invasive techniques, we cannot capture the entire brain. So maybe the solution is fusing different kind of modalities. And if yes, you think the same, what should be the first step, you think?
Well, I mean, if I had the choice, I would use single unit recordings from all 86 billion neurons in the human brain if I could get that information. I mean, I'd be very happy if I could get that non-invasively from healthy participants. And I mean, we can't. And I think the problem is that there's not just a difference in technique, but there's also a difference in species. So if you record one kind of signal in a healthy human and the invasive recordings are from whatever, a monkey, I mean, I'm really not against monkey research. I've always defended and said that we need this kind of research because we need to understand how the brain works. We wouldn't understand it based on fMRI alone. Yeah, I mean, the basic understanding of the basic mechanisms in the human brain is done by animal research. I want to acknowledge that. But I think when we come to cognition, I think we need to think about potential species differences as well. So if you study consciousness in a monkey or in a human, there might be some different things going on there. So I'm not saying not to do it in the monkeys. What I'm saying is that when you have to kind of look at differences, you might have to kind of think about the species barrier as well. So, I'd say, I mean, I don't have a good answer what I would be interested in studying. I mean, what I'd like is at least something like voltage-sensitive dye imaging in human brains, and that doesn't exist. But I think that would be something that would give you the resolution in space and time. I think we could get quite far, at least with cortical studies, with that kind of level of resolution. but I don't see how we could get that. And so, and I've seen too many cases where in neuroscience, we've been distracted by looking at the same brain area again and again and again and again and again. And I mean, like, for example, the surprise that you can reversibly inactivate LIP, the area that's supposedly summing up all the information during motion decision making, and then you can reversibly inactivate it, and the monkeys don't even change their behavior. And you think, well, that's supposedly the integration area, but if you reversibly inactivate it, there's no difference in the behavior. So, but that's where kind of like people have had assumptions about putting the electrodes. So it's, perhaps we should also be a bit more open-minded about looking elsewhere in the brain.
Okay, unfortunately, it's already past 4 p.m. now, so we will need to shut down this Zoom meeting now. Thanks everyone for participating, and a really big thank you to John for
thanks for the invitation. Yeah, sorry for the disruption earlier on.
No worries.
Okay.